Shall we begin? Welcome. Now we are beginning our first unit, that is the genetic material. Now you have already studied all the content related to the genetic material. So because of that, you know that what is genetic material. You know very well about this term genetic material. We simply call nucleic acid in that particularly the DNA as the genetic material. So how this DNA was discovered as a genetic material, that we have to study in this unit, genetic material. Okay, now the first point in this genetic material unit, first is the what is genetic material. Now those substances which act as a genetic material, that can be defined as the substance that carries the information determining the properties of that organism and responsible for transferring genetic information from parent to progeny. The simple meaning of this definition is that whatever the information that is possessed by this genetic material, okay, that is possessed by that genetic material, contains all the information which is needed for the cell to remain live condition. Okay, so whatever the enzymes they are needed for that various pathways, that information is present in the genetic material. Even in the in case of cell division, whatever the enzymes, whatever the factors that are required, that are present in this genetic material. Similarly, this genetic material also has the ability to transfer genetic material from one generation to the next generation, that is from parent to progeny. So we get all this genetic information which we have in our body in today's condition, that is derived from our parent. So that information carried from the parent to us in the form of DNA. So those substances which are acting as a genetic material, that substances must possess the property, that information which is required for that organism to survive. Second property which is needed there, that information must be transferred from one generation to the next generation. Okay, that should be transferred. So all these characters, they are satisfied by those substances, they act as a genetic material. Now we know that in almost all the organism, the genetic material is the DNA molecule. Means right from the bacteria to the higher case, blue whale, even elephant. In this organism, the genetic material, even in the human beings, the genetic material is the DNA. But there are a few exceptions are there. In that, the exceptions are the few bacteriophages and numerous plants and animal viruses, in which the genetic material is RNA instead of DNA. Okay, so some of the organisms are there which carries RNA as a genetic material, but most of the organisms, they contain DNA as the genetic material. Okay, but how this genetic material was discovered? Earlier, it was believed that, you know, that our body contains highest amount of protein. Okay, highest amount of protein. So because of that, earlier it was thought that the protein which is present in every living organism may be the genetic material. Okay, it may be the genetic material, but because of some characteristic which is possessed by that protein molecule, it is considered as a genetic material instead of DNA and RNA. So what are the properties which are possessed by these proteins? Okay, all the known intracellular molecules, only the protein were the sufficiently complex and variable in structure and in chemical properties to contain the great variety of information required for the genetic substance. So one property which is satisfied there, it should be complex and it should have variable structure and chemical composition. You know that the protein is composed of various 20 amino acids are there, which are arranged in different position to get different type of proteins. So similarly, these proteins, they have different structures like primary structure, secondary structure, tertiary structure. So that's why it is considered that the protein may be the genetic material of living organism instead of DNA. Now, what DNA is considered at that time? DNA was considered at that time as a possible genetic material because it was known to present only in the nuclei of the cell. Okay, DNA wasn't considered as a genetic material because it is only present in the nuclei of the cell, not present in the cytoplasm. So that's why DNA wasn't considered as a genetic material. Another point which is considered there, why DNA is not considered as a genetic material, because it is too simple to be a gene and was assumed instead to be a structural component of the chromosome. So earlier DNA was considered as only the structural component of the chromosome. Now you know that the DNA while there is a division in eukaryotic cell occurs, the nucleic acid, that is the DNA material, gets himself coiled around the histone proteins to become the chromosomes. So earlier it was considered that DNA was a simple gene, and it is instead of that, it is a structural component of the chromosome. So DNA wasn't considered as a genetic material at that time because of these reasons. So now there are some experiments which are carried out, and these experiments, they are simple, which are done with great care, and these experiments then identified DNA as the genetic material. Now the first point in this unit, it is the evidences for DNA as the genetic material. So there are 3 experiments are there which are carried out in a simple manner but with a great care, which identified DNA as a genetic material, not the protein, not the carbohydrate, not the lipids. Okay, now in that first experiment, which is carried out by specifically a scientist, that is Fred Griffith, and that experiment we called as a transformation experiment as well as Griffith experiment because it was firstly discovered by the scientist Fred Griffith. Now the current idea of the development of the current idea that the DNA is a genetic material developed with the observation in 1928 by Fred Griffith, who was studying a bacteria responsible for human pneumonia. That bacteria we call Streptococcus pneumoniae or pneumococcus. This bacteria able to cause in humans pneumonia. While studying this disease in human beings, this scientist made some observations, and through which it concluded that there is some factor is there that we are going to see here. Now the virulence of this bacteria was because of the surrounding polysaccharide capsule. Okay, now this pneumococcus bacteria, it contains a capsule which is made up of polysaccharide which surrounds the bacteria, and because of this capsule, this bacteria get protected from the defense system of the human beings. Okay, and when we grow this bacteria Streptococcus pneumoniae on the agar medium, it produces smooth edged colonies on agar surface. Okay, it was known that mice were normally killed by smooth bacteria. So this Streptococcus pneumoniae bacteria, which is carrying this surrounding polysaccharide, and that polysaccharide protect it from defense system of the body, and it produces smooth edged colonies on the agar surface. That's why this bacteria, it is particularly denoted as S type bacteria. Okay, so this bacteria is able to cause the pneumonia disease in mice. Okay, so while studying this, this is the Streptococcus pneumoniae bacteria under the after the staining. You can observe here Streptococcus means cocci which are present in chain, and this bacteria produces Streptococcus pneumoniae bacteria produces smooth edged colonies on the medium. Now similarly, Griffith also isolated a strain of Streptococcus pneumoniae which is having a rough edge colonies, and that it is called as a colony mutant. And this rough-edged colony, rough-edged Streptococcus pneumoniae bacteria, which is to be proved both non-capsulated and non-lethal to the mice. Means this rough-edged bacteria, it is not able to cause pneumonia disease to the mice, as well as when we cultivate this rough-edged bacteria on the medium, they are non-capsulated. So while conducting this experiment is divided into three different steps. Firstly, Griffith injected mouse with live rough bacteria, where he observed that the mouse wasn't infected. See here, this is the mouse which is infected with live R cells, live Streptococcus pneumoniae bacteria. When it is injected with this bacteria, that mice remain healthy, means it is not affected by the pneumonia, so it remains as it is healthy. In second experiment, Griffith injected heat killed smooth Streptococcus pneumoniae bacteria in a healthy mice. You can observe here, this is the Streptococcus pneumoniae bacteria which is having a capsule. This is the rough edged bacteria which is without a capsule. When this healthy mice was injected with heat killed smooth cells, then these mice get also remain healthy, not suffered from the pneumonia. So what it indicates that already we have seen that this yes bacteria is capable of causing pneumonia to the mice, but heat killed yes bacteria it is not able to cause pneumonia in the mice. Why? Because the bacteria get destroyed by heat killing. So this mice remains healthy. But in another experiment, you can see here this live S bacteria when injected in a mice. You can see here this mice get suffered from pneumonia and then it died. But in another experiment where he injected the healthy mice with live R cell, live Streptococcus pneumoniae bacteria, live R cell bacteria, a mixture of these two, live R cell plus heat killed S cell bacteria. You can see here, when this mixture injected into a healthy mice, okay, that mice get suffered from pneumonia and died. When the bacteria was isolated from these killed mice, it was observed that that bacteria is of live S cell type. So how this happened? Already we have seen that heat killed S bacteria, it is not capable of causing pneumonia to the mice. Live R bacteria, it is also not capable of causing pneumonia to the mice. But how this happened? This live R bacteria and heat killed S bacteria, when this mixture was injected in a human, in a mice, that mice suffered from pneumonia and after some time it get died. How this happened? And when we observed that, when we isolated bacteria from this killed mice, that was of S type, live S type. How this happened? This happened because there is you can observe that when we kill a bacteria by heating, whatever the genetic material that is present inside that bacterial cell that get released outside the bacteria, and some of that part, the genes which are responsible for causing the smooth nature, that is transferred from heat killed S cell to the live R cell bacteria. So we can able to observe some kind of transformation. Here the killed bacteria is of S type, but the live one is the R. Somehow this S gene is transferred to the R cell and transform this R cell into a live S cell bacteria. So same thing happened here. Same is there. Now several years later, it was observed that when a mixture of R and heat kill S bacteria were grown in a cultural fluid, S living celled bacteria also produced. In a lab, when we grow this bacteria, both a mixture of live R bacteria and heat killed S bacteria, when we grow in a cultural fluid, live S celled bacteria were also produced. A possible explanation for this surprising phenomena is that the R cells restored the viability of dead S cell bacteria. But the idea eliminated by observation that the living S cells, when we grow, even when the heat killed S culture is in the mixture, was replaced by the cell extract. Means that instead of using live S cell, living S cell, and after killing that, whatever by using the centrifuge machine, we are going to separate the other components of the bacteria. We can only keep the cell extract. When we use the cell extract of broken S cell, and that used, same type of results are also observed, where our bacteria is get converted into a S cell bacteria. It was concluded that somehow this R cell bacteria, which is present in a live condition, is get transformed into a S cell bacteria, and that condition where there is a transformation of live R bacteria into live S bacteria, that Griffith called it as a transforming principle. Okay, so in this experiment we saw that this some genes, they are transferred from killed S bacteria to live R bacteria, and this transfer of genes from S to live R bacteria, when this live bacteria then get converted into a live S bacteria, and this live S bacteria is capable of producing capsular polysaccharide, and this capsular polysaccharide, because of that, this S bacteria, it is lethal, causing the pneumonia in that mice, and because of that pneumonia, that mice get died. So here you observe that this experiment concluded that there is something is there which is transferred from killed S bacterial cell to the live R bacterial cell, and converting this live R bacterial cell into the live S bacterial cell. So he concluded that there is something is there which is changing this live R bacteria into a live S bacteria, and he called that as a transforming principle, the nature of which is unknown, whether that is protein, whether that is carbohydrate, whether that is DNA, whether that is RNA. The nature of that, it is unknown for the Griffith. So this is the beginning of the experiment, where it is observed that there is the change occurs of R bacteria into the live R bacteria, conversion of live R bacteria into the live S bacteria, and some of this is occurred because of some transforming principle. Something is transferred from S to killed S bacteria to live R bacteria, and changing that live R bacteria into a S live bacteria. And then another experiment is carried out by the 3 scientists after 15 years of the Griffith experiment, which is carried out by 3 scientists, that is Oswald Avery, Colin MacLeod, and Maclean McCreary. These three scientists, they partially purified the transforming principle from the cell extract and demonstrated that the transfer that something which is transferred from killed S bacteria to the live R bacteria, that was the DNA. Okay. Now these workers modified known scheme for isolating DNA and prepared samples of DNA from S bacteria. That hit killed S bacteria, from that the DNA sample was extracted, and that then this DNA sample was added that DNA sample was then added into live R bacterial cell, allowed to react after a period of time. They placed a sample of S containing R bacterial cell on agar surfaces and allowed to grow to form the colonies. When such experiment they have carried out, they observed that some of the colonies, about 1 in 10 raised to 4, that grew were of S type. Means in lab we can also carry out this experiment where something is transferred from killed organism to the live organism and changing that live organism to the killed type of nature. So to show this was a permanent genetic change, they dispersed many of the newly S colonies and placed them on a second agar surface, and resulting colonies were again S type. Whatever the colonies that they got after this experiment, where they mixed the DNA of S killed bacteria in a live or bacterial culture and allowing them to react and then plating on the agar medium, they found that some of the colonies, they are of S type. When they dispersed these S bacterial colonies and placed on second agar, they observed that these dispersed colonies were again of S type. Similarly, whatever the R colonies they grown on this agar medium, they also disperse them and subsequently transferred on the new agar surface, and these R colonies also retain the R character, whereas transformed S colonies also retain the S character. So because R and S and R colonies, they differed only in the presence of a capsule, that is polysaccharide coat, which is present around the S bacteria. The ability of purified polysaccharide to transform was also tested. So the difference between these two bacteria is that the smooth bacteria, they have the capsule which is composed of polysaccharide, and rough bacteria, it is without capsule, it doesn't have the polysaccharide. So only difference between these two is the presence of polysaccharide. So they purified the polysaccharide and used to transform the bacterial cell. So when this purified polysaccharide from S bacteria was purified and used to transform the R bacteria, such here in this experiment was no transformation was observed. Okay, so during this there is no transformation was observed. Similarly, they also carried out 4 different procedures to show that the genetic material is nothing but the DNA. Firstly, the chemical analysis of the sample containing the transforming principle showed that the major component of that chemically, when this hit killed extract was analyzed, that showed that the major component was a deoxyribose containing nucleic acid. Second observation which they made, physical measurement showed that the sample contained highly viscous substance having the properties of DNA. Okay, highly viscous substance containing the properties of the DNA. When we extract the DNA from the bacteria, we observe that during this, the nature of this DNA is the highly viscous material, which is like that of the properties like that of the DNA. Similarly, they also carried out some experiment to demonstrate the transforming activity, whether it is lost or retained, by using highly purified proteolytic enzymes such as trypsin, chymotrypsin, a mixture of both. Similarly, ribonuclease enzyme, an enzyme that depolymerize RNA. So this shows that the active principle was neither a protein or nor a DNA. Nor RNA. Now, treatment with material known to containing DNA depolymerizing activity, it inactivated the transforming principle. So, from this experiment, they concluded that from this experiment, every MacLeod and McCarthy, they drawn a conclusion that the DNA was a genetic material. They drawn a conclusion by saying that the nucleic acid, that is the DNA, has the biological specificity, but the chemical nature of that transforming principle is yet to be underdetermined. You can see here same experiment where the mix R cell with DNA extract, and same experiment when R type is allowed to grow on the medium, no transformation occur. Type R cells, type S DNA extract, transformation occurred. Type R cell, type yes DNA extract, DNase enzyme. This DNase enzyme destroys the DNA molecule, so no transformation occurred. Similarly, type R cells and type S cell DNA extract and RNA enzyme is there added there, transformation observed. Similarly, types live R cell and type S DNA extract protease enzyme is also added there. Here protein, and that is protein is destroyed, whatever the protein is present in this DNA, that is S type of DNA extract, but transformation occurred there. So you can also observe a simple video which is shown in this two experiments.
This is our timeline of the history of genetics. So far, we've covered the important milestones up to this point. Yes, we've taken a look at the work of Gregor Mendel, followed by the work of Sutton and Boveri. This was the chromosomal theory of inheritance. Later, it was Thomas Hunt Morgan who put forth his work on the common fruit fly and achieved another milestone. Now comes the next one. After Morgan gave an experimental evidence of the fact that genes for the traits are present on the chromosomes, the scientific world then got together to find out what exactly makes up these genes and chromosomes. In other words, the chemical nature of this genetic material is what everyone was curious to know about. In the same era, a British bacteriologist was busy handling his pneumococcal strains. They are nothing but the bacteria named Streptococcus pneumoniae, which are the causative agents of pneumococcal infections. Frederick Griffith found out that there is some factor that helps transform one bacterial type into the other. Let's have a look at this remarkable experiment. To begin with, we need to get introduced to the two types of pneumococcal strains. This one is a smooth strain denoted as the S strain, and this one is a rough strain denoted as the R strain. Why are they named so? This smooth strain, as we can see, has a smooth outer surface. It's the capsule around the cell wall which protects it from various threats. The presence of this capsule makes the strain virulent or strongly infectious, to be precise. The absence of such a smooth coat makes the strain avirulent or non-infectious, to be precise. Now you may ask, why is the R strain avirulent? It's the absence of the capsule which makes it susceptible to the immune cells of the host's body. Now getting back to Griffith's work, let's see what experiment he exactly performed. He first injected a mouse with the S strain, which is the smooth strain of the pneumococci. As expected, the mouse died. Similarly, on injecting another mouse with a rough strain, the mouse survived. This was pretty much anticipated by him because the rough strain is usually the avirulent one. What was done next? Now the third mouse was injected with the S strain, but after the bacteria were heat killed. That means he heated the S strain to extreme temperatures, which killed the virulent strain and made it ineffective. So the injected mouse survived. Now comes the last part. This time, Griffith injected a mouse with a mixture of both rough strain and heat-killed smooth strain. That's right, a mixture of the rough strain and the heat-killed smooth strain. Now, what do you think would have happened? Most of us would expect the mouse to survive because both are individually ineffective or avirulent. However, to his astonishment, the mouse did not survive. How did this happen? And what do you think Griffith concluded? With this, he concluded that there is something from the S strain that was passed on to the R strain, resulting it to turn virulent. Also, do you know what he recovered from the body of the mouse? He recovered live S strain. Surprising, isn't it? This further confirmed that the R strain was transformed into the virulent S strain. He named the transforming factor as the transforming principle. He did not really know what exactly this was and hence named it just the transforming principle. But he predicted that this could be the genetic material. Was this really the genetic material? Or was it something else? Let's find that out in the next part. Imagine we're given a bag full of flowers, and we are told that there is one flower which has the Midas effect. Anything that the flower touches gets transformed into gold. But how will we find out which flower is the special one? All the flowers look more or less the same. Well, the only thing we can do is make all the flowers touch some object. Isn't that right? So examining every individual flower is the only option we have. The story was similar in the case of transforming principle. After Griffith concluded his experiment on the bacteria named Streptococcus pneumoniae, the science world was now curious to know more about the transforming principle. Finding the answers was not a piece of cake though. It was almost 2 decades later that 3 scientists figured out what this transforming principle was. They tried finding out its chemical nature by testing every single component of the virulent bacterial strain. Let's move a step ahead in the timeline and understand the interesting experiment performed by Avery, MacLeod and McCarthy around the year 1944. The 3 scientists were quite fascinated with Griffith's work. Just like others, even they were curious to know what the transforming principle was. So they thought of continuing the same experiment with better research. Griffith concluded his experiment by inferring that there is some factor that's passed from heat-killed smooth strain to the rough strain, which is avirulent. This factor transforms the avirulent strain to the virulent strain. So the 3 scientists thought of planning the experiment by targeting the heat-killed smooth strain. Just like a bag filled with flowers, the smooth strain cells had various chemical components in them. And just like how we had to examine the touch of individual flowers, the scientists mixed every single chemical component individually with the rough strain. So let's say this smooth strain cell had these 4 components A, B, C and D in it. They mixed the component A with the rough strain and injected it in a live mouse. Similarly, they mixed all the other components individually with the rough strains and injected the mixtures in different mice. What they found at the end was only one of those components among all was effective in transforming the rough strain. It was the case in which the mouse died. The conclusion was that component D transformed the R strain. They isolated this component from the heat-killed strain and tested it further. On chemical analysis, they could figure out that the molecules had a chemical composition similar to that of DNA. Deoxyribonucleic acid, abbreviated as DNA, was known to people by this time, but it was not given much importance. Nobody assumed that it could be such a vital part of the cell. In fact, people thought that proteins made up the cells. And it was also believed that proteins make up the genetic material. So this experiment was a huge breakthrough in genetics. Many tests were carried out to investigate this further, and the chemical composition of this transforming principle was found. It had components in the ratio that were found in the DNA molecules. So to further understand its nature, this component was tested using chemicals that break down the constituents. For example, certain enzymes like protease degrade proteins. Use of such chemicals help us identify the constituents in a given sample, as they will degrade specific molecules only. So in this case, the chemicals used to degrade DNA were used along with others, and it was found that the chemicals successfully broke down the molecules. It implied that the molecules were that of DNA. This chemical analysis helped in understanding that the transforming principle was nothing else but DNA. And this wasn't easily accepted as a fact. A few years later, another experiment helped confirm the fact that it's indeed the DNA which is transforming principle. Let's find out more about the experiment.